This protocol is an extremely low-cost method of collecting data on organismal physiology in an accessible and standardized way. This tool can be used to explore physiology at several levels. It can be employed by intrepid field biologists as well as young school children. The main advantage of this technique is that it relies on readily available materials and is very low cost. It can be performed nearly anywhere and by researchers with a range of expertise and goals. This method can provide insight into physiological, behavioral, and ecological processes. As the key metrics for eliciting a physiological response are standardized, the method can increase our ability to understand and compare key traits across groups of individuals, species, and ecological communities. Handling insects without damaging them takes practice, but practice makes perfect. Defining the parameters of your experiment, like how long to keep insects in ice water, can take time, and it requires experimentation and understanding of your focal taxa. Demonstrating the procedure with me will be Heather Rohr and Matt Standridge, two technicians from the Daniels Lab.
Begin with identifying the species of interest, keeping in mind that each group will differ in the time it takes to induce a chill coma. Based on the organism and use of data, choose different cutoff points for the experiment if the focal individual does not fly. Conduct a pretrial on a small sample to determine the key parameters like time required on ice to induce a chill coma. Choose a cutoff time based on the ecology of the species, keeping in mind that after many minutes of being incapable of flight, some insects are taken by predators. Collect insects using appropriate methods such as baited traps and entomological nets. Place each individual in a separate